colony that was founded by one initial bacterium. Because they reproduce asexually, we can have a founder that consists of one. And we want a founding one of one. All right, so we'll keep doing that to make sure we get that. Then we'll do PCR in it. So we're going to try to amplify those two specific genes in separate reactions. We'll run them out in gels to see if it worked. If we know that it worked and we have the DNA, we'll use the gels to quantitate. How much of a yield did we get? Because then we'll set up a sequencing reaction. So what we're going to do then is we're going to take that sample, we're going to quantitate it, and we will then send it out for sequencing. So we're going to do two genes, 16S ribosome RNA gene and RPOB gene. Those are two targets. We'll go over that. Hopefully I'll have enough time. So we should end up with large quantities of that. We quantitate it, we send it out for sequencing, and then we'll get sequence back from a sequencing lab. And then you're going to take that sequence and submit it to a database. And they're going to search all the sequences that have been submitted to that database, which is billions of sequences, and they'll try to match it up to specific species. I have no idea what species you'll get, but more than likely it will be a genus of Vibrio. Not necessarily, but more than likely because of the way we're doing this, and we're going to have colonies that are bioluminescent. So we're using kind of a criterion to proceed through here that will sort of focus on this genus. Okay? Although we have occasionally had other genera that are bioluminescent. Okay? We'll learn about photosynthesis, the capture of light energy and conversion into chemical energy. Very critical for all ecosystems. We'll study complementation experiment, which is really nifty, easy set of experiments. We'll do them 3 consecutive labs. So again, the reason why it's important to work with the same samples each week is because you'll process those for 3 weeks, and in the end, you look at the data, and you can figure out the number of genes in a pathway. And we're not doing it, but you could easily figure out the order of the pathway. So you could very easily say, I know nothing about this pathway, but I can generate some mutants, analyze these mutants in a matter of some weeks, and almost pretty much know the entire pathway. It's an amazing technique, very powerful. So you'll be working with complementation for 3 experiments. You'll learn about mitosis and meiosis. The DNA is sent out for sequencing. When we get that back, we're going to then blast it, so we'll be doing what's called bioinformatics. So a lot of looking at what that species is, how does this species compare to the other species found in the lab, what are the relationships, which ones are more closely related, et cetera, okay? All from the sequence. And we'll work with Mendelian genetics, classical fruit fly stuff. So that's part 1. That will be the basis of lab exam 1 for 100 points. Any questions? We sort of call those the wet labs. The second half of the labs do involve dissections. Next week, I'm going to talk a little bit about animal ethics, why we use the animals, et cetera. But we will dissect a rat in the vertebrate anatomy lab. We're going to ask you not to name them, okay? We're going to ask you to treat them with respect. And what happens here, and I think it's really important for you to understand, is we first have you dissect the rats, and then we will donate them to an organization that must use rats to feed the wildlife that they rehabilitate. So they have to purchase the rats if we don't purchase them for them. So what we're doing is a step in between that process where you learn from them, okay? The only difference is if we weren't doing it, they could go out and buy rats that cost $2, but the rats we get are pathogen-free from specific labs, very specific protocols, and we pay $20 a rat. But that's to make sure that there should be pathogen-free, okay? So we'll talk about that more when we do that, okay? But anyway, that's what we do. It's a little expensive. We spend almost $7,000, $8,000 on rats. You're worth it, okay? Invertebrate 1. For invert 1, we'll do some more dissections. We will dissect cockroach, worm, the animals of clam, starfish. You'll dissect a crayfish, or things like that. When we get there, we'll do it, okay? We have a lab on reproduction development. So you'll actually take sea urchin egg. Hopefully, it'll be sea urchin egg and sperm. Mix them. See a fertilization event. See the sperm penetrate the egg. What happens as a result? You'll see cell division, various things like that. You'll look at various stages, okay? And so when I say dissections are mandatory, I should probably put a little asterisk there. We're not going to force you to do the dissections, okay? But we are going to ask that you take that lab exam too, which is a common set of questions for every student. So if you're really opposed to doing the dissections, you can come talk to me. Usually what I say is there's almost always someone very eager to do the dissections. So if you have 2 people who are partners and neither one of you want to do it, see if you can split up for those eager beavers who really want to do it, okay? And what happens in this class is quite interesting, is we get a set of students who just love the first part and just hate the second, and vice versa, just love this part and hate this, and then we have those who love and those who hate, okay? We'll try to make it as interesting as possible, okay? And we'll have a lab exam that it's a lot easier now that it's all students take the same exam. The stress level is much more bearable, okay? We used to have a paramedic on call. Just kidding. No paramedic on call. But when we get to the sections, if you're feeling faint, we have had students faint, sight of blood, various things like that. So we do a blood pressure measurement. I'll talk to you about safety. Safety is important. The GSIs will do this more in small groups. They can point out various things to you. But if you're ever feeling sick, and we've had to call 911 on several occasions. We have had fires in our lab support space. So when the alarm gets sounded, we do have to evacuate because the fire could be next door, which in a couple cases it was. The ceiling caught on fire. So just watch out. We do have to evacuate. Cell theory. We'll talk more about this next week because we're going to have a lab on this. Dr. Down has introduced it. I mentioned it briefly. Hierarchy. You're pretty familiar with this. You know, molecules, macromolecules, organelles, cells. You know, tissues. Tissues are cells that have a common function, then organs, organ systems, etc. So we'll go over all that. Evolution, can't stress it enough, okay? But evolution is typically not occurring in an individual. It's occurring in a species or population, okay? And I think that's key to understand. Development is different than evolution. Development is for an actual individual. Evolution is different. Classification, 3 domains. I'm going to put this on the board because I'm even confused by it. And I always lose chalk. And it always ends up at the computer. Okay, so here's what I'm putting on the board. When I was a biology student, well, I'm still a biology student, okay? I'm still learning. But back when I was in college, we had stone tablets. No other tablets, but no, seriously, we talked about bacteria, and we talked about eukaryotes. And what we said was eukaryotes are those things that have a membrane-bound organelle that contains DNA. Okay? And the literature reflects that for 200 years, this concept of bacteria. But 30 years ago or so, people realized that really we should talk about prokaryotes. And those are the ones that lack that membrane-bound organelle with the DNA. I'm not going to membrane-bound, I'll be specific. Membrane-bound organelle called the nucleus. But what we found was there were 2 groups in here. There was a group that we said were archaeans. And they're actually more closely related to the eukaryotes than they are to this other group, which we call eubacteria. But you can see why this is really confusing. Because historically, the term bacteria, because we didn't know about this group, people use this. So when you look at old literature, they talk about bacteria. They're not specifying between these 2 groups because they didn't know about this group. So we use the term prokaryotes with 2 domains. The 2 domains are eubacteria and archaea. Sometimes people are really sloppy, just say bacteria here and archaea here. I try not to do that. I want to realize that I'm not trying to use this old historical term of bacteria, so I try to use the term eubacteria. Is that clear? So you have to be a little careful when people talk about bacteria. I have one HMI professor, well respected. When he talks about bacteria, he calls them bugs. So I think most people think of insects. So clearly not the choice of words. So I will try to be very careful when I use words. If you think I misspoke, feel free to say, did you mean this, okay? Likewise, during office hours, if I correct you, it's not to be mean, it's trying to make sure you understand so that when you go home, you don't say, well, I said bacteria, but should I have said you bacteria? I will probably correct you just so that you don't have that confusion later, okay? It's not to be mean, it's just to help you learn, okay? Structure, function are related. I mean, it's interesting. Things come back trend-wise, glasses, clothes, whatever. The same true with biology. 40 years ago, structure and function are highly related. And then that started with on the bi-side, people kind of ignored it until we got new tools. Now that we have new tools like X-ray crystallography, things like that, we learn a lot more about structure, and then we can look at intimate function, okay? So now that we have these tools, we're going back to structure-function relationships a lot. Ploidy. I'm going to do this now. It's okay if you don't understand it. I think you'll be able to repeat the definition, but maybe not apply it. Ploidy refers to the number of sets of chromosomes. If you could write that down, that would be great. Ploidy, sets of chromosomes. It took me 6 years to get the Campbell book and the other biology books to correct ploidy. They were all using it wrong. I had one professor, it took me 5 years, individual meetings, before he finally said, but I published papers in these fields. And I said, but I don't edit them. And if I did, I'd change them. He now understands it, okay? Ploidy, sets of chromosomes. So in us, male or female, we have two sets in most of our cells. One set, of course, came from our mom, and one set came from our dad. And that your mom got one set from who? Her mom, her dad, right? So she had two sets. Effectively, you have a set that came from your great, it came from Adam or Eve, right? Effectively, you can work backwards with changes. So we're all related, it's just how distantly related are we, okay? But sets are actual sets. So in us, we have 46 chromosomes in our somatic cells. There are two sets, each of 23. So there is chromosome number 1, the biggest, chromosome number 2, the next biggest, 3, etc. And they come in pairs because they're in sets, okay? The way it should be done to keep it straight is if you see 1n equals 23, this number is the number of chromosomes in this number of sets. So it's 2n equals 46, means there are a total of 46 chromosomes in those 2n sets, which means there would be 23 types. Okay? And I'm hoping Dr. Patel sort of uses a similar system. A lot of times I have professors say humans are 23n or 46n. And that's clearly not the case. We don't have 46 chromosomes, 46 sets or 23 sets. What they mean is 1n equals 23 or 2n equals 46. So keep that in the back of your mind. I will ask a lab exam question for sure on that. Okay, so let's just do something. Anyone know sort of what they say the number of genes in the human genome? 30,000. To make it a little easier to do the math, let's say there are 23,000. Okay, 23,000 genes. So in your diploid cells, how many genes do you have? 23 or 46,000? Those say 23,000, raise your hand. Only two. Those say 46,000. Okay, about 20. And the other 500 people? No genes. There are 23,000 genes in a set. So if you have two sets, you still only have 23,000 genes, but you have two versions of each gene. So you haven't changed the number of genes. You just changed the allele numbers, right? And if those two alleles were the same in those two sets, what's the term we use? Homozygous. And if those two alleles were different, heterozygous. That introduces a lot of genetics right there, okay? Safety, common sense, your GSIs will go over this. You will need to sign a safety sheet that says you're aware of it. Now, we are not going to require you to wear safety glasses. We'll provide them if you want. We are going to ask you to tie back, I don't have this problem, long hair when you work with open flames. There has been one time a student caught their hair on fire. They weren't quite sure why I was patting their head down. They eventually smelled it. Other students came over to do the same thing. So tie back long hair. You must wear shoes. You can't ride your bike in lab. You can't roller skate or skateboard from one side of the room to the other, okay? And along these lines, if you forget your bike lock, that doesn't mean we can put your bike in the lab room blocking the exit, okay? Sorry, you'd have to leave it outside as a donation, because that's what it would be. Go home quickly or something, okay? So please make sure you wear those safety things. Micropipetter, they're shown here. They work great. We have 3 sets. We'll actually have 4 sets. A P1000, which is for volumes up from like 200 microliters to 1,000 microliters or a mil. So that's P1000. We have a P200 for about volumes 20 to 200. We have a P20 for volumes about 1 to 20. Then we'll actually have P2s, which are for volumes like 0.1 to 2. So those are very precise if you think about it, okay? The way they work is as follows. I'm going to do this demo. The GSIs are going to do it again. Always use a tip. Always use a tip. And I say this because we have had as many as 90% of the micropipetters contaminated in a lab. I'm not sure why, but the way we're going to get the tips is as follows. And you guys are going to be very good at this. Who I have to correct typically are the GSIs at the GSI meeting, which is after this. When you want to take the tip, rotate it open, grab your tip. If you want to double seat it, that's fine, and close it again. Every time this is open, any bacterial spores, fungal spores, anything in the air can be contaminating those tips. So you really want to be careful. So we're going to use the sterile technique. Here's the micropipetter here. Here's where you set the volume is this window here. You can see the window. Well, okay. Andy, thank you. Sometimes projectionist doesn't do this. So you see the window here. That's for the volume. Here's the plunger. There's 2 points of resistance. I'm going to move it slowly. There's that point. But if you notice, I can push my thumb past that. This is the point that corresponds to the volume you set. So if you want to remove some solution from a tube, you'll put the tip in there, and you will slowly release that plunger. And then to dispense it, what you'll do is put the tip typically on the side of the tube. And I know you can't see this. You'll do this on the side of the tube because capillary action helps wicks away the liquid. You push down in the plunger. Now, as you can see, if I can zoom it, there's no liquid left. But I have a little reserve movement on that plunger. And we'll have waste containers in the lab. I don't have one here. Where you can take and move this plunger past that point to eject any residual liquid, okay? Again, the first point of resistance is the key one. To remove the tip, there's an ejector button here, and you just push on it and the tip will come off, okay? So always use a tip, and remember that first resistance, okay? And we have 3 different volumes. And what you're going to do as exercises is just weigh them, okay? Weigh the samples you take out. There's a balance there, very quick. I think you've used balances before. There'll be some plastic weigh boats here. I see students trying to drop the liquid on the weigh boat from the top. I can understand. We actually try to tape this off so you can't do that. So if you see things taped in place, that usually means don't adjust them, okay? So we take that, add your sample from the side. If you're really nervous, especially when I walk around, people get really nervous. I'm there in the lab to see if you have questions. I try to interact with you, etc. It's not like I hear people say, oh, guys here. It's not there to scare you. If you're really shaky, put your elbow to balance, okay? It's fine, all right? Anyway, I know a lot of you have used these before, but good technique is going to be really important. When we do that PCR, we'd love to have it work, okay? So anyway, the fast way to weigh these out, add your sample, record the weight, tare it, back to 0. Add your sample, tear it. Add your sample, tear it, right? So real quick, you don't need to dump this out every time or get a new wayload every time. Just add your sample, weigh it, tear it back to 0. Is that clear? It goes fast. We only have 2 balances in the room. There'll be 28 students, 14 pairs. We usually work in pairs. For this, you'll be working in pairs. So you're going to walk over to the balance. We're going to start with station 14 and station 1, different sides of the room, avoid bottlenecks. 14 will work first, 1. When 1's done, they're going to tell 2 to start. 14 is going to tell 13 to start, then 12 until we meet in the middle, okay? And it's important to do it that way because it's a bottleneck. We only have 2 sets of balances for each room. So I don't want everyone at the end of the lab to say, oh, I still haven't done this yet, okay? Does that make sense? The other thing that happened is, for the specs, which is the next part of this, we'll go over the theory of it, but the specs connect to computers. Apple told us the order was 2 to 3 days. Once we placed the order, Apple said, oops, we meant 2 to 3 weeks. So we don't have the specs right now. They should come on Wednesday or Thursday. Sorry, the computers. We don't have them yet. So we're going to have a few groups share a computer. So group 1 is doing the balance, group 2 can use that computer, and then if group 14 is doing it, group 13 can use it. We'll avoid all bottlenecks, okay? So we have, we'd like to have 14 per room. We only have 9. So a few groups have to share. So again, doing that system will minimize that bottleneck. I apologize for that. We placed the order a while ago. Should have been here last week. They kept saying it will be, and then they finally messed up, okay? The way the specs are going to work is there's a light source. There are various ways. This is how some specs work. There's a prism that diffracts in a specific wavelength. There's a slit so that wavelength, light of a certain wavelength goes through your sample. Then there's a photodetector that creates some voltage that gets measured and converted into a readout, whether it's transmittance or OD. Okay? And this just shows you the old spec that we had. We don't use these anymore, but I want to point out to you one thing. You can see that there's a relationship between transmittance and absorbance. And it's the minus log of transmittance is the absorbance. Okay? Minus log of transmittance is the absorbance. It's in the lab manual. The new specs, it's all computers. All you see is a number. So when I found out last semester when I asked this question in the lab exam, students did a lot worse on that question this semester than in the past when we used this equipment. Because when you use this equipment, you see every time you take a reading that direct relationship. With a new system, you never see that. So that's why I'm trying to stress it now. Okay? It's just something lost by the system. Here's the system we use. We'll put the sample here. It will blank. What it literally does is because it interfaces with the computer, it has the light source, and then it will do, as it goes through the sample, it will diffract it in the wavelength, and then there's a CCD array here of the various wavelengths, and it converts how many photons are there to a basically absorbance and transmittance. You can select which value you want for that wavelength, and it can scan the entire range from, let's say, I don't know, can scan the range from 340 nanometers up to 700 in about half a second. So it's real quick. So when you put your blank in there, it will take and do some values for that. And the blank is going to be the background contribution in your sample. So we have a red dye. That red dye is dissolved in water, so what should the blank be? Water. If that dye was dissolved in acetone, what should the blank be? Acetone, because it's always the background. So what happens with the computer is when you put that water in there and it blanks it, it takes those OD values at each wavelength, keeps track of that. When you put your sample and say do the reading, it takes the OD, or transmittance, depending on what you want, and it automatically subtracts out the blank for every wavelength, and it's done. Again, half a second. So you can take these readings very quickly. In the past, we had to go very slow with the old specs. So it works really fast. You'll get an absorption spectrum that looks like this. And as you know from chemistry, different compounds will have different absorption spectra, or NMR spectrum. And that's because they have different arrangements of bonds, electrons, functional groups, etc., right? So clearly you know that you can do identification using the spec, right? You've done that in OChem, probably done it in GeoChem. And of course you can do it for quantification. Because if you know the molar extinction coefficient, you can solve some equations, or you can just look at a standard curve like this. So in lab, we'll provide you with a standard curve. What your exercise will be to blanket, put your sample in, create an absorption spectrum, and then figure out the wavelength at which you have maximum absorption. That's the wavelength we use for the standard curve. And that kind of makes sense, right? Because if you take the wavelength that has the maximum absorbance for the smallest change in concentration of that substance, it will give you the largest change in OD, right? So that's the lambda that you sometimes hear about. So you got to figure out the wavelength and then use the standard curve for concentration. Is that clear about the specs? So you can clearly use the concentration, use the standard curve, or solve an equation, and identification. Now, I'm not going to give you an absorption spectrum and say identify this compound, okay? But what I could do is I could give you an absorption spectrum like this and not have the label say ABC, and I could ask you which one might appear more bluish. It appears more bluish if it's, what, absorbing it or not absorbing it? Not absorbing it. So when you see green leaves, that's because green is not absorbed, okay? This is what the lab will sort of look like. There's the spec there. There's the computer there. This is for the enzyme lab, which will be later, but we'll be doing catalysis. We'll have specs and the specs and the computer set up. And normally we have 2 per table on each side. We have to share a little bit more. The other big part of this lab is the microscope of this. Learning how to use the microscope. And why do you even want to learn how to use it? Because with a microscope, you can magnify images, right? And that allows you to see better resolution. What is resolution? Resolution is defined as the ability to identify 2 distinct points of light as distinct points. So if this microscope has that resolution, and if I put them closer, I can't see them, and this is microscope A, this is microscope B, and they can be closer together, and I still see that there's 2 distinct points, this has a better resolution, a smaller D value. And it's a smaller value. So when we say a light microscope has a resolution of 0.2 micrometers, and then we say electron microscope has a resolution of 20 nanometers, the 20 nanometers is a smaller value. It's a better resolution. Sometimes people say it's a better resolving power, okay? So that's what resolution is, the ability to see those at 2 distinct points. Okay, so with our microscopes, I'll give you a moment to write that down. We're going to do a few exercises. Okay? We're going to use these microscopes throughout several labs, and we're going to try to measure the sizes of various things. So we need to have some measuring device that we know that we can use to measure things. So one exercise is to calibrate a measuring device. The measuring device is called a micrometer. I've learned it introduced too much confusion, so let's call it a ruler or measuring device. What happens is, in the ocular, so there's 2 sets of lenses, oculars and objectives. The objective lenses are closest to the object, which is on the stage. So the object's on the stage. Just like with the spec, when we